Welcome to the Rare Plant Monitoring Program training. This training will be broken into numerous videos that walk volunteers through the process of preparing for a rare plant survey, conducting the survey, and submitting data. This video will discuss Level 2 protocols for estimating large plant populations during rare plant surveys. Using Level 2 protocols is not required to participate in the Rare Plant Monitoring Program, but they're available for anyone looking to collect more statistically rigorous data. Level 2 survey protocol is a more accurate way to estimate large plant populations. Whereas the Level 1 protocols are a quick and dirty method that is easy to implement and adaptable to surveyors with various time constraints, the Level 2 protocols are more involved. Level 1 protocols can't be used for statistical analysis without substantial uncertainty, but the Level 2 protocols can. They are intended for volunteers looking for a more rigorous way to detect change in a population through annual or semi-annual monitoring. A step-by-step worksheet is posted on the Rare Plant Monitoring Program website under the Volunteer tab, then Forms and Guides. It's important to establish a clear objective before conducting formal sampling so you're confident the question you're interested in answering, in this case, how big is the rare plant population, can be answered by the data you collect. The objective of the Level 2 sampling is to be 95% confident that our estimate of the plant population is within 20% of its true size. Because different plant populations are distributed differently, the same number of quadrats cannot be used to sample all populations. Let's look at a plant population that is distributed uniformly across a site. We would only need to sample from a few quadrats to get an accurate estimate of this population size because each quadrat would have the same number of plants in it. In fact, a single quadrat is a good representation of this population because the variation among quadrats is very low. Now let's look at a population that is distributed unevenly. In this case, the variation among quadrats is high. Some of the quadrats sampled would have lots of plants and others would have none. No single quadrat is representative of the entire population. In order to get a good estimate of the whole population, we will have to sample a number of quadrats in this patchy population. So how do we figure out how many quadrats we need to sample from? In order to answer this, we need to know something about how the plants in the population are distributed. Is it uniform or patchy? So before your formal survey can begin, you will have to do what is called pilot sampling to determine what the correct number of quadrats to sample is. The purpose of pilot sampling is to get a sense for how the plants are distributed in space and how much variation there will be between the number of plants in each quadrat. To do your pilot sampling, simply meander through the population and sample a few 1 meter by 1 meter quadrats. Count all the plants in each quadrat and record them on a sheet of paper. Next, calculate the mean and standard deviation for these pilot sampling quadrats. These formulas are in the Level 2 worksheet in the program manual. To calculate the mean, add up the number of plants you found in each quadrat and divide that total number by the number of quadrats you sampled. Calculating the standard deviation, or the variation between quadrats, takes a few more steps. First, subtract the mean number we just calculated from the number of plants in each quadrat. Then square this number. Next, calculate the mean of these squared differences. Finally, take the square root of this mean. This is the standard deviation. You can also calculate both the mean and standard deviation using Microsoft Excel. The mean is calculated using the average formula, and the standard deviation is calculated using the formula shown here. Next, use this equation and the results from your pilot sampling to determine how many quadrats to sample during your formal survey. In the equation, n is the number of quadrats you should sample during your formal survey. S is the standard deviation. And B is 0.2 times the sample mean. In our example, the average number of plants per quadrat from our pilot sampling was 3.4. So B would be equal to 0.2 times 3.4, which is 0.68. If we plug in the numbers from our pilot sampling, we would get this equation. We should sample 26 quadrats for this population. Next, you need to determine where your quadrats will be located. The first step is to measure the length and width of the population. Whichever number is longer is your baseline, and the shorter side is your sideline. In this example, the baseline is 11 meters long, and the sideline is 7 meters long. Next, select a random number between 1 and the length of your baseline. In this example, that's a random number between 1 and 11. Do the same for the sideline. In our example, we would select a random number between 1 and 7. Starting from one end of the baseline, walk to the random number between 1 and 11. Then walk perpendicular to the baseline, the number of meters you selected between 1 and 7. Set your first quadrat here. If the random number we selected for our baseline was 3, and the random number for our sideline was 5, we should walk 3 meters down our baseline and 5 meters up our sideline and lay the quadrat down. Set up the quadrat with one corner at your foot, one corner closest to the start of the baseline, and the other two corners toward the start of the sideline. In other words, the quadrat should be between the second and third meter on the baseline and the fourth and fifth meters on the sideline. All the individuals of the target rare plant population are counted. I make separate tallies for the seedlings, flowering individuals, fruiting individuals, and vegetative individuals. After finishing counting all the individuals of the target species in quadrat 1, head back to the junction of the baseline and the sideline and repeat the process 25 more times. Here's the layout of the 26 quadrats over the rare plant population. After tallying up all the seedlings and vegetative, flowering, and fruiting adults, here are the numbers from the example formal survey. Remember that although the entire rare plant population occupies 77 square meters, we only sampled 26 1 square meter quadrats, or 26 square meters total. To get an estimate for the entire population, we need to multiply our totals by roughly 3, or actually 77 divided by 26, which is 2.96. These numbers can now be entered into the data collection form. We are 95% confident that these estimates are within 20% of the true rare plant population size. The Level 2 protocols are clearly more labor-intensive than either the Level 1 protocols or visually estimating the population size. On the other hand, they will result in a much more accurate and dependable description of the population. That means that the status assessments and management decisions made based on these estimates can be done so more confidently. For volunteers who are interested in taking their monitoring to the next level, this could be a good option. It may also be a good option for volunteers looking to monitor the same large population annually or semi-annually. Volunteers may be interested in small research projects like studying the effects of a management technique or annual rainfall on a rare plant population. In these cases, a more rigorous sampling technique would be best for answering those questions and sharing the results with others. For more information on possible research projects like these, or to discuss the Level 2 survey methods, feel free to contact the program coordinator. This concludes the Level 2 survey protocol video. Please continue watching the remaining videos to complete the rare plant monitoring program training requirements.